Hello listeners and welcome to the Dr. GPCR Podcast. Did you hear about the Dr. GPCR University? It's the ultimate learning platform within the Dr. GPCR ecosystem. Here you can expand your knowledge, connect with experts, and stay informed about the ever-evolving world of GPCR research. We offer both live and on-demand courses covering everything from GPCR pharmacology data analysis to drug discovery strategies. Our courses, like those that focus on using Prism, help you master the pharmacology data analysis framework, navigate the complexities of GPCR research, and improve your work's quality, reliability, and efficiency. Join us and elevate your GPCR expertise today. And now let's dive into this episode.
Hello, everyone. This is Yamina from Dr. GPCR. Welcome to another episode of the Dr. GPCR Podcast. This very early morning, I'm very happy to have with me Benjamin Clements. Ben, welcome.
Hi, Yamina. I'm really excited to be here. As we were talking before, so excited to be alongside some of the great early career investigators you've had and some of the titans of the field that you've had on the podcast. Happy to add my 2 cents to everything.
Very excited to have you on. I think it's important to be as inclusive as possible on the podcast. So a big shout out to you and all of the recent early career scientists who we've invited and who accepted the invitation. As I was mentioning before we hit record, it's been a very difficult path to get more early career and junior scientists on the podcast. But it's important to make your voice heard. And as you were mentioning in the beginning, you might end up having your episode released right before somebody who's been in the field or after somebody who's been in the field for 20, 25 years. And you can actually brag about it and say, hey, I was on the same podcast as so-and-so. All right, so let's start at the beginning. Could you please introduce yourselves, tell us who you are and what is it that you do?
Yeah, so I'm Ben Clements. I'm a postdoctoral fellow at the University of Michigan. I've really only been in the GPCR field for about 2 years now. I'm more trained as a traditional drug development scientist. Started my career at the University of North Carolina. Didn't really know exactly what I wanted to do scientifically. I knew I wanted to be a scientist. I was one of those kids that wanted to be an astronaut way too late in life and then realized that I had to be in much better shape than I am to be an astronaut. But I knew I wanted to do science. My father is an engineer. My mother's a teacher. My sister ended up becoming a veterinarian. My brother, an engineer. So big science family. And I didn't know what science I wanted to do for the longest time. So I got involved with research at the University of North Carolina, studying the transport of choline across the intestinal epithelium in a research lab at the UNC Eshelman School of Pharmacy. And that's what really sparked my interest in, okay, we can do chemistry, we can do biology, but we can do it in a way that is applicable. This is relatable to patients. It's relatable to people. This can help someone at the end of the day. It's not just raw basic science. And that's something that I locked onto. And so I chose to pursue my graduate education at the University of Minnesota, where I studied with George Wilcox and Carolyn Fairbanks. Looking primarily at ion channel inhibition and the pharmacokinetics, pharmacodynamics of small molecule inhibition of the NMDA receptor. And so that got me interested particularly in using pharmacotherapies to treat chronic pain and opioid use disorder, which pushed me now to my postgraduate education, where I'm working with John Trainer here at the University of Michigan, where I'm looking at primarily opioid pharmacology, looking at positive allosteric modulation of the opioid receptor as a way to treat chronic pain, to enhance the analgesia components of opioids, the benefits, the powerful pain relieving effects, while limiting those negative side effects, the respiratory depression, the constipation, the abuse liability. And so far, we've had a lot of success with that in our preclinical assessment and continuing to try to develop new molecules and figure out exactly how these allosteric modulators are inducing the changes on the opioid receptor to create these impressive in vitro and in vivo effects.
I love it. Sounds like you definitely are coming from a science-based family, but it also sounds like as you were getting into where you are today, there was a more and more focus on not only the drug discovery aspects of it, but also membrane proteins.
Mm-hmm.
So
yeah, absolutely.
Ion channels, And now the real deal. I'm kidding. Well, this is the Dr. GPCR podcast. We cannot, it's not the Ion Channel podcast. So we cannot, we have to be biased towards GPCRs.
Oh, no, I absolutely love GPCRs, particularly as a drug target. They've been, I mean, so many drugs target GPCRs, and this whole, like, allosteric modulation has existed for a while in ion channel fields. You have the benzodiazepines, you have the barbiturates, but allosteric modulation as a clinical tool in GPCRs is so novel and so interesting, and there's so much availability. Like, there's been some work, particularly from Arthur Christophilos's group, on the muscarinic acetylcholine receptors and a bunch of other GPCRs that people are starting to investigate, but it's so new and so exciting from a drug development standpoint. And it's sort of, I fell into drug discovery, then I fell into neuroscience, and now I've fallen into GPCRs and sort of hitting my stride in terms of the combination of effects.
Yeah, there were these companies dedicated to identifying allosteric modulators for GPCRs out there. So there's a lot of work. I feel like it's challenging because you need to have a good pharmacology understanding. You need to be running the right assays in the right settings in order to tease out that property.
Yeah, that's one difficulty that we've had, particularly in my training. I'm originally trained as a pharmaceutical scientist. I had done my preliminary pharmacology testing, but I hadn't really dug into like the mathematics that underlie a lot of the concepts in pharmacology. And so that's one thing I've been learning, along with the membrane biology of GPCRs, really understanding their function and their mechanism outside of a purely pharmacodynamic perspective. And so that's been one thing that's been really exciting. And one benefit of working at the University of Michigan is we have such great people in both of those fields that have been able to learn from and really start to tease those things apart. That's one of the big benefits I've seen with my postgraduate education, is I get the freedom to just spend 2 days reading pure math about pharmacology of allosteric modulators. And then I could spend 2 days just looking at crystal structures of the mu-opioid receptor and interrogating that. And it's been a lot of fun.
Yeah. And let me guess whose paper is you're reading on pure math. I don't think I need to guess that. Everybody knows that it's most likely Terry's papers and with Arthur and the entire, molecular pharmacology teams. How did you end up at the University of Michigan? What drove you from a career perspective?
Yeah, so I'm interested in pursuing an academic position following my postgraduate education. And I was sitting with my advisors at University of Minnesota, and we were talking about what we needed. And I was interviewing at a couple places and really trying to decide what benefits I would have from each program. So I was looking at more raw neuroscience programs at like University of the Medical College of Wisconsin. I was investigating some more like neuroscience drug development components on the West Coast. And really what it came down to was thinking about, okay, what are you really passionate about? What kind of programs would you want to get an academic position in? And I started thinking, well, I'd like to be in potentially a medical school or pharmacy school. I really love to teach. I like training professional students. I like training PhD students. And so they said, okay, getting a core in pharmacology, like that actual, the basics of just the old school pharmacology. Like I love all these new techniques with the RNA scope and the really complex systems, but I needed that basic understanding because everything is built off of that. If you don't understand the math that underlies an allosteric modulator, it doesn't matter how many cool, fun tools you have. You're not going to really understand what's happening. And so that's what drove me to Michigan. They were like, okay, you have these great professors there that can teach you all these things and that have a track record of producing just outstanding scientists.
Yeah.
And so that's one thing that really drove me there.
The other question I had for you is from a human and mentorship perspective and team perspective. What drove you to your current lab? What did you like about it? What were the guiding principles that got you to where you are by saying, okay, this is what I want because X, Y, Z?
Mm-hmm. Yeah. So in terms of the lab perspective, of course, from the top down, I wanted people with extensive mentoring experience with particularly training academics. Coming from a pharmaceutical perspective, I've worked with a lot of people that have trained industry people, but not as many people that have 3 or 4 students that have gone on for long-term professorships.
Yeah.
So I wanted someone from top down, I wanted someone that had that kind of experience, particularly extensive funding experience, and I could get that training face to face. From bottom up, I am a really strong proponent of that science is fun. Like science is great. And so I wanted people that I could get along with, that I could joke around with, that I could still produce great science. But like for the other day, I worked with John Trainer and he created the GTP gamma S assay. And we got into a long argument about what would happen if you gave someone a intravenous dose of GTP gamma S. Like what would happen? We talked about that for like 15 minutes and it was great. It was just absolutely irrelevant to anything we were doing. But it was a fun scientific conversation that we got to have just for the sake of it. And it was I really loved that experience. I worked with a lot of really great people. And I admire the scientists that are able to work in these labs that are really intense, where you just put your head down and focus and get your work done. And I'm sure people really thrive in that environment. I needed something where I could have those conversations and that collaboration.
Yeah, I love it. I have to mention, you mentioned having fun and science is fun. I was mentioning your shirts today, which I think is really beautiful and colorful, especially on this relatively cold day. I'm assuming it's a trend within the lab as well. So we work very hard, but at the same time, we have a lot of fun.
Exactly. Yeah. We come in, we put our head down. Every day we put up like a trivia question on the board. One of our other graduate students, Kat Demery, absolutely amazing scientist. I've seen her work harder than a lot of people I've worked with. And she really loves squirrels. And this month is Squirrel Appreciation Month, apparently. So she writes a trivia question about squirrels every single day on our whiteboard, right next to all of our dose response curves and our mechanistic diagrams as we puzzle things out. And so, yeah, we try to keep things engaging and collaborative. And I think that really helps build our science, at least the way that we do it, because everything that we do requires us working together. If something starts to go wrong on one of our assays, if we're running 400 plates of GTP gamma S with 20 or 30 different agonists, and our EC50s start to shift on our standards, we have to be able to communicate that. We have to be able to talk about that. And we have to have those systems in place. And so I find that having that fun collaborative environment allows you to just bounce off each other very efficiently.
And you made a great point about, running 400 plates and being able to look at the standard. It's important to be able to have a system where you have more than one person looking at it and you have a way to visualize the data. Because in my experience, looking at just numbers, fitted curves, you have to get a sense of the curve. You have to look at it. You have to get a feel for it. Is it a steep curve? Is it not a steep curve? And what did the raw data look like? All of these things become very important in order to then get to conclusions and then move forward in your program or in your assays.
Yeah, and I think along with that, one thing that's really helped me, and particularly at the Michigan environment, a lot of programs have it. Some programs don't because of the way that the university is structured. I've been able to learn a lot more about the basics of pharmacology and understanding it because we have a great undergraduate program. So we have a bunch of undergraduates that work in our lab that through Michigan, they have the undergraduate programs where they can apply. They provide us funding and support to bring on. I think right now we have 7 or 8 different undergraduates working in our lab. And so I get to meet people that have never done a concentration response, who are learning these assays. Maybe they've touched a pipette, and I have to, in my own head, come back and say, okay, what really underlies a concentration response curve? What drives this effect? I can't just say agonist, antagonist, competitive, non-competitive, uncompetitive, and expect them to understand it. I have to know it so that I can put it forward. And that's really helped me think about my science in new perspectives. Like, it's wonderful to be around these great high level long-term scientists, similar to the podcast. It's you talk to these people that have been doing amazing work for 20, 30 years, but then you talk to someone who's brand new and they'll give you a whole new idea because they're coming at it with a fresh perspective, a new idea, a diversity of thoughts. And that's been absolutely wonderful.
I agree. And I think the younger generation is lucky in the sense that we had these trailblazers, but at the same time, methodologies and techniques have evolved. You know, I wasn't in that let's say age group where you would do PCRs by manually taking your tube from one water bath to another with different temperatures. But I was in the cohort where you couldn't run GS cyclic AMP assays in a plate reader. You had to use radioactivity and then purify your cyclic AMP, your tritiated cyclic AMP produced or inhibited, whatever you were looking at, on columns, and then quantify your radioactivity, which I think is you get a specific appreciation to the fact that you can just run a plate and you have everything on the plate and way more conditions that can be tested on a plate for sure. And I'm just talking about here 96 well plates, not even getting to the 384 well plates where you can get even richer information and also time course information, which I think is very important for us to understand how receptors function.
Oh, yeah, absolutely. And that's one other benefit of sort of the program that I chose. One of the reasons I chose to come here is that I get to learn these pretty straightforward, kind of old school assays like GTP gamma S, radioligand binding. We've done some cyclic AMP, some beta arrestin recruitment. Right now, we've been pushing into more of Brett based assays, so a little bit more of modern, quote unquote. But understanding those really classical assays and going back to papers that were published in the 80s and the 90s and being like, okay, there was a reason these choices were made, and building that into these advanced techniques has been really beneficial. Thinking about like, okay, well, we have this receptor, we have these compounds. I could put it in a complex system where I'm looking at maybe live brain imaging. We have the resources to do that at Michigan. It's great. It's wonderful. Can I process that information on a basic scale? And so, yeah, we've been doing a lot of these really old school traditional assays that are well validated, well understood, and allowing that to build into more complicated processes.
Yeah. No, I agree with you. It's funny because I didn't mean to say that the old school stuff isn't good enough, because at the end of the day, you can have the fanciest assay and the most high throughput assay, but if the data is confusing, going back to GTP gamma S or radioligand binding is the truth. Does the thing bind or not?
Yeah, absolutely. And it's fun that we see all of this stuff in like these. I always tell students, it's like you see these papers where they're putting out these incredible assays with these really complex systems. But I know in these labs, maybe they're not publishing it as forwardly as these really cool systems. They're validating everything on the basics. They've either run these in the past or they've put them through. It's like, yeah, it doesn't matter if you're doing this really complicated Brett system with all these different components. They probably ran some sort of binding at first because you have to know if it binds.
Yeah. And at least you hope so.
Yeah. At least a lot of the big papers that I see come out, I know that they've done at least the basics before, and it's important that you have to have the basics in place. And that again comes back to training the undergraduates. It makes me go back over the basics every single day. Yeah. I can't just say, oh, run GTP gamma S with this, and give me this, because I have to explain exactly what I'm doing, what my thought process is, and they get to push back on me, because sometimes I'll say something and be like, wait, why am I doing that? That doesn't make sense. You're absolutely right. That's just the way that I've been doing it. Yeah. So I get to reassess my own basics every single day, because with science, you're growing and you're building all the time.
No, absolutely. Let's talk a little bit about the project that you're currently working on. Obviously, if you're uncomfortable revealing details, I won't push, but I'm curious as to what is it that you're working on right now, and why do you find it exciting?
Yeah, luckily, a lot of the stuff I'm working on, we do have some stuff that's a little bit more protected, but luckily everything I'm doing is pretty available. Is that I came in with a very pharmacokinetic, pharmacodynamic background, in vivo pharmacology primarily, and then some mass spec as well. I had not done really much in vitro at all. And so I came in, basically we have these positive allosteric modulators of the opioid receptor. We've shown consistently in preclinical models that they'll enhance the antinociception in the warm water tail withdrawal assay, the hot plate assay, but they hadn't really been tested in chronic pain models because opioids are already very effective for acute pain. But what happens if we put it in chronic pain, the people that might have to take it consistently, can we understand what's happening in that population and better apply it? So I've been performing studies seeing how these modulators can enhance opioid analgesia in neuropathic and inflammatory pain states, collaborating with one of our colleagues in plastic surgery who has this really interesting neuroma model. Neuropathic pain is less responsive to opioids than other types of pain. Neuroma is completely unresponsive in patients to opioids. It's really sedation. It's the only benefit they get from them. So we've been investigating to see, can these modulators make opioids effective in neuroma? And preliminary data seems to suggest, yes, it can, which is really, really powerful. And so we have that's our sort of in vivo component, putting these in chronic pain models and seeing, can we recapitulate what we see in the acute models? Secondarily, we have these allosteric modulators, and we're just curious, okay, we have these odd behavioral effects. We have this difference in effects. Exactly how do these compounds even work on the level of the receptor? We know they bind the receptor. We know they can enhance in vitro. Exactly where are they binding and how are they eliciting their response? Yeah. So we're doing some it's been absolutely wonderful, just old school pharmacology in vitro, doing synergy assays, doing antagonism assays, and doing site-directed mutagenesis to see if we can enhance or suppress the allosteric activity by changing certain amino acids on the receptor. And one thing I didn't mention is that we're looking at primarily two classes of positive allosteric modulator, two very chemically distinct classes, a thiazolidine and a xanthine-dione. They don't look anything alike. They are very unique, but they do the same thing. And we think they might be binding at a similar site on the receptor, or at least eliciting similar responses. So we want to interrogate that. And then because we're at Michigan, we have all these resources for drug development, take all that information in vivo, in vitro, compile it all, send it off to chemists and say, okay, we believe that making a structure that looks like this will create a new chemical entity that we can develop as a therapeutic and make a druggable compound to help patients. Because that's what I'm particularly interested in. Is it's wonderful to figure out this biology of GPCRs, and I admire people that are really interested in doing that because that's what everything is built on. I'm curious as to, okay, can we take all of that amazing information, smash it together, make a drug?
Yeah, no, I agree. I like it. I particularly like the fact that you have two classes of molecules, two different scaffolds that do the quote unquote the same thing, which gets me to my next question. Do you have any plans or any capabilities for cryo-EM? Do you want to see? I'm assuming the answer is yes, but how will you get to see where your molecules sit? The assumption, well, I don't know, these are allosteric modulators. They can be sitting at different places and doing similar things to the receptor.
Yeah, absolutely. So we don't have the resources to do cryo-EM in our group. We've been collaborating a lot with different groups that have the resources to do this cryo-EM. But the compounds that we have are not particularly potent, and there hasn't been a lot of success of getting these onto a crystal or onto a cryo-EM. And so there was one group in Japan that was able to put out a proposed structure with one of the compounds. And really what they had to do is they combine cryo-EM with NMR and have a proposed site on the receptor, which is one that we've been investigating in more pharmacological systems to see, okay, can we use this site to change the activity of the allosteric modulator? But really, it's been incredibly difficult to get these, as it is with a lot of allosteric modulators. They just don't bind well enough to get really great images the same way you can with agonists and antagonists.
No, for sure. We'll see if they're not potent enough. you may not get there. Also, in order to see them, or at least to get there halfway, you'd need to have the cryo-EM with a native ligand in order to enhance potentially that binding, or at least decrease the energy barrier to get into that active state.
Yeah, absolutely. And additionally, the proposed site that has been put forward, I'm no expert at cryo-EM, so maybe I'm completely off base here, but because the binding site is proposed to be within the plasma membrane, it sort of adds that additional little barrier that doing a GPCR, getting a crystal, getting a cryo-EM, you already have to deal with the fact that it is a membrane-bound receptor. And then if you're integrating things into the membrane, then it becomes just additional steps.
Yeah, no, it's interesting that you mentioned that. So there is this small molecule for a class B receptor, and there's a structure associated with it. And it turned out that it's an allosteric, and it binds next to the membrane under the receptor. Which I thought was a very interesting place to be binding. And whenever you discover allosteric modulators, you think that it's somewhere on the receptor. It is, but at the same time, it's in my mind, it's outside, somewhere outside, not embedded or under there.
Yeah, that's one fascinating thing of allosterics from a drug development standpoint is it's not as clean as this conserved, evolutionarily devised pocket that is built in. Like we work with opioid receptors, so like mu delta kappa, we have difficulty finding things that are highly specific for one because you have this really conserved pocket. Yeah. But allosterics, it could just be anywhere. You've made something that happens to interact with the receptor, and then there's proposed endogenous allosteric modulators, but they're not as well characterized. They're certainly not as evolutionarily driven as a agonist pocket.
Yeah. No, agreed. So then my assumption is that your favorite GPCR or GPCRs are opioid receptors.
Oh yeah, I love the opioid receptors. I mean, mu is my favorite. I think I'm a little bit out of order that I think I love kappa the second most because people don't give kappa as much love as I think it deserves. There's great researchers who really love kappa, and I'm happy that they do it. It's more complicated than a lot of the other receptors, so people tend to just it goes third for a lot of people. But especially from a drug development standpoint, the mu-opioid receptor, like you have this, basically pharmacology was built on the mu-opioid receptor. Like earliest drugs that were developed were poppy extracts. Yeah. And you have this huge history. The characterization of morphine was a huge step in chemistry. The characterization of the opioid receptor was a huge step in molecular biology. And it's this incredibly powerful tool for treating patients that we've used for millennia. And although it has this incredible negative side effect, it will still probably be relevant for years and years and years. But it's also, we have these incredibly powerful benefits, but it's also incredibly dangerous. They're risky. They're not the safest. You have to be careful with them. And so it creates really a perfect encapsulation of drug development, is that you have to think about the good, but you also have to think about what could this do to a patient. They're not the safest molecules, and you have to be cautious with them. And so, the mu-opioid receptor is it's so fascinating. It's endlessly complex, but still beautifully simple at the end of the day.
Well, I feel like if something comes off as simple, it's extremely complex. It's really like, I took many of Terry Kanakin's courses, and the way Terry explains pharmacology, it sounds so simple, but at the same time, it is not. And you can tell that he's mastered it to a point where he can think of it he can explain it in a very simple manner, but we all know that it's not.
Mm-hmm.
So,
absolutely. Yeah.
I love it. And plus, what I also wanted to mention is that you're right. You are in let's say a Great Lakes GPCR pharmacology hub there at the University of Michigan with so many fantastic researchers.
Yeah, it's been absolutely wonderful being able to work at the University of Michigan. We have our bi-weekly seminars where we have visiting faculty. We have our student seminars, and just being able to have these conversations with John Trainer, Alan Smirka, the opioid people in the Domino Center, Erica Levitt, Will Birdsong. It's been absolutely amazing to have just being able just to watch them have discussions, not even participate myself, but to watch them think about things has been just absolutely brilliant. And that's, I can't, I recommend it highly, but there's also so many other great programs that exist in the same way. So if you can have the opportunity to spend a little bit of time with these researchers that have been around for years and years and doing such great work, I can't recommend it highly enough.
Well, you're right. And I think in that sense, and I was talking to Ian Cronis earlier, and I was mentioning that here in the US, this rotation system is really fantastic because you get a chance to try out different things with no strings attached when you think about it. Back in the day when I was doing my master's and PhD at the University of Montreal, they didn't have that. You picked the lab and you were there. And if you didn't like it, you had to, it was a difficult thing to extract yourself from a place and then find another one. How do you justify switching? Whereas you have the opportunity to do rotations, you get to see different labs, different teams, different PIs, and then you kind of figure out which shoe fits.
Mm-hmm. Yeah. So when I did my PhD, I was a direct admit. I went from undergraduate. I was brought into Carolyn Fairbanks' group in the Department of Pharmaceutics, and the rotations did not exist in our department. It was a small department with kind of like isolated cores of people who were doing raw chemistry, people who were doing the physics of like tablet formation, and then a small core of people who were doing neuropharmacology, which is what I was interested in. And so we didn't have the opportunity, which I was very lucky in that I fell into a lab that I really meshed with, that it was a lab that, as we talked about before, that was able to have fun, but do great science, to put our head down and focus, but also to have fun conversations, joke around. This was in the period of Game of Thrones. So I believe Friday morning or Monday morning, because it came out on Sundays, we would have a long discussion about the newest episode of Game of Thrones. And it was a great lab experience where I got exactly what I wanted out of it. But I am very lucky in that. I'm very privileged because I walked into a lab that fit, as opposed to people that I never had to deal with, mentors that I didn't get along with. I never had to deal with science that I didn't love. And so I feel like it's very difficult in those sort of environments. And that's one reason I really love the rotation system, is back to what we were talking about earlier. Not only do you get to learn techniques, you get to kind of play with tools that you might not have ever played with. You get to see the people as they actually are. You're not seeing what they do in invitation week, where they put on their best face and they show you all the great things of the university. You get to see what a day-to-day activity is like. And so I'm really in favor of that kind of rotation system. And the people that we've had come through our lab as rotation students, they'll come in for 3 months. They'll do great science. They might get on a paper if they pick up the skill quick enough, and it's wonderful.
Yeah, no, I agree. All right, top 3 aha moments that you had so far in your career.
My whole career.
Well, yeah, anything that you feel like, oh, I learned something and now I think differently because of it, or I made a decision based on that experience that led you to where you are today.
Yeah, I have a lot of them. I was thinking about this earlier, and it was one thing that I was chatting with some people about is sort of a more first is like 2 kind of career little development ones, as opposed to just raw science, is that when I'll do one undergrad, grad, and then post-grad. So when I was an undergrad, I was just sort of learning the techniques. I was sort of finding my footing as a scientist. And we were having a lot of difficulty because we were doing radio-labeled tracer studies across in vitro cultures. And we were having a lot of difficulty because we were dealing with radioligands and we were dealing with biohazard. And so we were having a lot of difficulty. And of course, the undergrad is the one that takes out the trash. That's how it worked in that lab. Yep. That's what I ended up doing. And we came to the odd decision of, well, we have biohazard stuff. We usually autoclave that and send it out for trash. We have radioligands. They can't handle biohazard. And I learned something really important in my career is I learned how to say no to a big name in a big field, because I had big names at UNC who were telling me, just put it in the autoclave. We don't want to deal with the issue. And I said, no, I'm not putting radioactive isotopes into the shared autoclave with everyone. And that was I was like, wait, yeah, I actually know things. My input is valuable. I'm an undergraduate. I'm 20 years old. I barely started, but I can contribute something to this group, and I have to be able to stand up for that. And so that was a big step. And I was like, wait, yes, I am a scientist. I have things to contribute. And that was really a big step for me. Another aha moment is sort of learning, again, coming along with me, figuring out who I was as a scientist, is I was in graduate school, and it came to the point where I had been doing oral gavage as a way to test oral bioavailability of a bunch of different compounds. And it came to the point where I was in this lab with these amazing, amazing behavioral pharmacologists, just doing incredible work. We had one lab technician, Kelly, who's just incredible behavioral pharmacologist, was able to do these types of injections that were incredibly complicated so efficiently. He had been doing it for 30, 40 years. And basically, the reason I got to grad school is Kelly's skill. And it got to the point where we were doing oral gavage, switching over to that behavioral paradigm. And I realized I was the only one in the lab that knew how to do it in rats. Mm-hmm. Now, I have a skill. I have a technical skill, something with my hands, not just my mind, but with my hands, that it makes me important. I was like, wait, yes, I am important to this team. And it was such a great experience to be like, wait, yes, I've had this imposter syndrome through my entire graduate career, but now I have something that's like, wait, this is something only I can do. I am necessary. I am important. And that was a big step, because I'm sure everyone who's listening to this has dealt with imposter syndrome at one point or another. If you haven't, I'm jealous. It's a nightmare.
Yeah, it is a nightmare. One thing I want to mention, so I was at an event here in the Boston area, and it was called Women Who Lead. And this question about imposter syndrome came up as well. So there were two panels, and the first panel with female CEOs who have the experience, and then the second panel was with female CEOs who are up and coming. And so the question about this came up, and so everybody responded. Turns out that 3 out of the 4 female CEOs had battled imposter syndrome, but the fourth one's answer was really interesting, because she was mentioning that actually she grew up with her dad is an ER doctor who actually put her to work, or there was something around training her, so she doesn't think about it. She just goes, because when you work in an ER and you have a patient who needs your help and your full attention, you don't have the time to think about it. So I thought it was an interesting little story that she was mentioning. But I think we all go through that, and it can be this, this circle, or this twisted way of getting lost in your head, or saying, oh, I'm not good enough, I'm not good enough, I'm not good enough, where actually I think everybody has it. I think everybody handles it differently. But the way I found to go about it is just put my head down and say, I'm going to try anyway. What's the worst that can happen? Because if you don't try, you're not going to get anything out of it.
Yeah, absolutely. And it's I think something that we deal with all sorts of personal struggles day to day in the lab, and at Michigan, there's always something going on. There's difficulties, whether that be imposter syndrome, whether that be stress about your dissertation. And I find that maybe it's something about I don't know, correlation versus causation. If it's something about people who choose to become graduate students, or something about the benefit of doing it, is I see people who are struggling, and most people just put their heads down and they get the work done. And it's something I really admire from a lot of people, that I see people going through really difficult times, and they are really strong, and they put their head down and get the work done, because I know that's what I do. And just seeing other people do it, it's just like, yeah, I think that's a really insightful thing, is that having the confidence in yourself and being able to just do the work is really important.
Awesome. All right. And number 3, I didn't mean to interrupt you.
No, totally all right. It's an important thing because I think it's something that affects everybody, and it's really important. And then I'll also do a scientific aha because I think that's what a lot more people will be interested in. And I think that comes down to our work with the neuromas. It's been really interesting because essentially we worked with Steve Kemp because he had the resources to do the spared nerve injuries that we do for our pain testing. Spared nerve injury, very standard nerve injury model that pretty much everyone uses. It's not particularly clinically translational because mononeuropathies are really rare in patients. And we were working with Steve. It's like, okay, you have the ability to do the spared nerve injury. You have plastic surgeons on your staff that can do these amazing surgeries and get that knocked out for us. We can do all the behavioral testing. And he said, well, we have these rats that have neuroma. We have this neuroma model that we primarily test as a way to look at nerve regeneration and different mechanistic things that they're building to try to improve neuroma in patients. And we thought, hey, you have these rats. Let's try them. Let's give them the drug. Let's see what happens. And we got an incredible response. We got a tenfold leftward shift in the dose response curve in methadone by the use of our modulator. And we were just blown away. We were just, how could this happen? We're running replicates just to see if it's true. But it was one thing that it just goes to show that you get the experience to just try things. One thing I love about academia, it's really nice being a postdoc. I don't have to write a dissertation. I can just try something. I could spend 3 days doing behavioral testing on these neuroma rats because we say that would be cool if it works. And we get to discover something that's incredible. And again, we need to prove it because this is science. We need to do our replicates. We need to do our mechanism to try to understand what's going on. But it seems to be really promising. And we get to, that all came about because we had a collaboration with someone who had a really cool system. And we said, hey, what if the peanut butter meets the jelly? What happens? And we found something absolutely incredible that probably will end up defining the early stages of my next career step, because we're trying to figure out, okay, if this is what's happening, can we put that into a grant that would potentially push towards a independent faculty position?
I love it. I love it. I was taking notes while you were talking for the team. All right, then any advice? I think we talked a little bit about it, but any advice for junior scientists who want to contribute to the field?
Yeah, I think we've been sort of hitting a little bits here and there on all of that. One big piece of advice that I really got that's really stuck with me, people who are listening that know me have probably heard this. Other people might have heard this. It was passed on from a graduate student in Minnesota, Kelsey Flepson, who pretty much day one gave me 2 big pieces of advice. Number 1, try to read a paper every single day. I pretty much only do work days. I'm good that I try to take the weekends. Try to read a paper every single day. Stay up to date on the field. It's really beneficial. And the big thing is academia is all about knowing when to walk and knowing when to run. So there will be weeks where just because of the way the experiments run out, you don't have that much to do. You can get some writing done, you get some reading done, but you need to take care of yourself. Maybe you only work 35, 36 hours that week. That's okay, because there will be weeks that come up where you work 60 hours because every experiment comes ready at the same time. Every animal's due. You have a grant coming up. You have a committee meeting. You have your dissertation. So you have to know when to preserve yourself and take care of yourself so that you can run when the time comes. And that's one thing I really took to heart. And it's like, yeah, sometimes I'll be sitting at work. I'll be there from 8 to 4 usually. 3 o'clock rolls around. I'm not doing anything productive. I have no experiments to run. My brain is fried from all the writing I've done that day. Sometimes I'll just say, okay, John, I'm going to duck out for the day because I know in 3 weeks I'll probably stay until 6 or 7 because something will come up that I will have to do. And it's important to keep that. It's not even just work-life balance. It's understanding that sometimes you have to take the time to take care of yourself because you know you're going to have to push yourself hard in the future. Yeah. And that was something Kelsey told me I took to heart very early in my graduate career and has probably saved me through all of this.
Beautiful. I think these 2 pieces of advice are fantastic, especially the second one. And I think it's not only applicable to academia, it's applicable to industry as well. You have to know when to walk and know when to run. And it's important. And, we're not, We're not machines. We need time to recharge. And the work that we do is not just technical. A lot of it is mental. So you need to be able to take a step back, take care of yourself, spend time with your loved ones, and the ideas will come. I remember during my PhD and postdoc, the best ideas came, in the shower or on a walk, because your brain needs time to process.
Yeah, everyone I've talked to that talks about these great ideas they've had, I pretty much hear two consistent things. They either thought of it in lab meeting or they thought of it in the shower. And those are the big places, apparently. Because if you're running an experiment, you're using your brain. You can't let those sort of abstract ideas come together. You need time to sit and just stare at data, to think about data, and to put it all together. And if you're constantly burning your brain, like your brain is a muscle. Technically it's not, but it acts like one. It gets tired. It needs a break. I come back from a long day of using my brain. Sometimes I want to put on the dumbest TV show I can imagine and just let my brain be off for 45 minutes before I get back to it. And that's okay.
No, I agree. All right. Last big question of the podcast. What should we title the episode?
Ooh, I feel like we've gone around a lot of places.
Yeah. I think, to be honest, you were mentioning the two pieces of advice that you got, and I think the number 2 should be the title.
I agree.
And I would change it to "Science is all about knowing when to walk and knowing when to run." What do you think?
I think that is perfect. Plus, then if people decide not to listen to the episode, which if you've gotten this far, congratulations. You get the big stinger right up front. You just see it come across your podcast app. You've seen it. Great, you've got that advice.
Yeah, exactly. And I think you're right. I think it's and we did cover a lot of ground, which is why it's very difficult to title these episodes. I used to do that in the beginning, then it became just too much, and then my team was saying, "Okay, Yamina, we need titles. Come on, we need some. You have really cool guests. We need something catchy." And I said, "You know what? How about we ask the guests?"
Well, it's the fun of we're all scientists. Yeah. Marketing is a very important skill that I am not the best at. And
it's a skill that just like confidence, it comes with practice.
Mm-hmm.
You have to be able to, and it's just like setting up an assay.
Mm-hmm.
And I bet you anything that anyone who's ever started working in the lab, on the first time where they were running or setting up an assay, even with guidance from somebody who's done it before, they felt uncomfortable until they did it a couple of times and then realized, oh, actually, now I get it.
Absolutely. Just hitting more great advice this week.
Yeah, exactly. Ben, thank you so much for your time today. I really enjoyed our conversation.
No, thank you so much for having me. This has been an absolute blast. Just a wonderful way to start my day. Now I can go into the lab feeling really inspired. And that's, I guess, one good thing about your program. And I've listened. I haven't listened to every episode, but I've listened to a bunch, and it's been just an absolutely great experience to have all of these different perspectives to help build up like the GPCR field. Because at the end of the day, we're, like you said at the very beginning, I talked about math. You can isolate that down to 5 or 6 labs. We're a small community, and it's great to have this resource. So I'm really honored to have been included.
Thank you. I really appreciate it. Good luck with everything.
Thank you.
I need a favor. If you enjoyed this episode, please click the like button. And if you like the podcast, consider subscribing to our YouTube channel. Many of you come back to watch our videos but aren't subscribers yet. One click goes a long way. All the information about Dr. GPCR is in the description. Find all our fantastic features and everything you can benefit from becoming a premium member. And while you're on it, check the Dr. GPCR University courses and share them with your network, colleagues, and friends. We also want to hear from you. Is there anything missing from our ecosystem? Can we make it better? Email Yamina at drgpcr.org or email our team at hello@drgpcr.org. Thank you so much for joining us in this podcast episode. If you're still there, we also want to take a minute to thank our team members: Yamina, Attila, Montserrat, Cam, John, Yatsu, Mark, Ivana, and Andreina. If you like our podcast, you can leave us a 5-star review on Apple Podcast. Until next time, and stay safe.